Hi and welcome to my OCR A-Level Biology revision with me, Christine. In today's lesson I'm going to look at saccharides, part of your Module 2 biological molecules. So the first thing you need to know is that there are different types of monosaccharides. So here we have our hexose monosaccharides. Now hexose means the 6-carbon monosaccharide. So a monosaccharide is basically a sugar, and you need to know how to distinguish between glucose molecules. So here I have 2 glucose molecules. Now they are only different in one place, but that completely changes the way that they will react. So it's important that we note that the first one on the left hand side is called our alpha glucose. Now it's called an alpha glucose because at carbon number 1 the hydrogen is above, therefore the hydroxyl group is underneath. If we look at the other glucose molecule, which is our beta glucose, the hydrogen at carbon number 1 is below, and therefore the hydroxyl group is the other way round. So it's important to note when you are remembering which one's the alpha. Alpha has its hydrogen above, beta has its hydrogen below. So that's how we distinguish between an alpha and a beta, which are our hexose monosaccharides, our 6-carbon sugars. Now we have pentose monosaccharide. So a pentose monosaccharide is a 5-carbon sugar. Now the difference between the ribose and the deoxyribose is again down to 1 part, and that's this time carbon number 2. So in a deoxyribose sugar, we have got 1 less oxygen. So the prefix DE, that tells you that something is being removed. So a deoxy, the oxygen has been removed at carbon number 2 of the ribose sugar, and that is what makes it a deoxyribose sugar instead of the ribose sugar, which has the hydroxyl group present. So that's a way to distinguish between your deoxyribose and your ribose, the pentose monosaccharides, and your 6-carbon hexose monosaccharides, for the ones that you specifically need to be able to identify, to draw potentially in the exam, and to show its structure. Now there's a few other hexose monosaccharides that you do need to also know, and they're important when we look at how they come together. So you also need to understand that there is a galactose, which is a hexose monosaccharide, and fructose, which is also a hexose monosaccharide. Now they will not expect you to draw them or to remember their structure, but they will give you potentially a picture of them and then ask you to pull some information out from that. So the important thing for us to understand about these hexose and pentose monosaccharides is that if they have a polar functional group, in this case the hydroxyl group, that will make the molecule soluble. So it's important that we note that our monosaccharides, our monomers of sugars, are actually polar molecules because they contain a polar functional group. Now, the polar functional group that we can see is actually our hydroxyl group. Now, if you remember, if you haven't watched already my video on water, then please do check that one out. But anything which has a hydroxyl group has a covalent bond between the oxygen and the hydrogen atom. That therefore is an uneven sharing of electrons because the electrons are closer to the oxygen than the hydrogen. Because the electrons are held closer to one of the atoms, what that does is that gives a polarity where one side is more positively charged and the other side is more negatively charged. And therefore, if there is a hydroxyl group present, that is going to make the molecule soluble because the hydroxyl polar functional group can actually form hydrogen bonds with water. So if we understand that, then how does the polarity change when we bring these monomers together? So here we have two glucose molecules, and what we want to do is we want to take these two monomers and we are going to synthesize a more complex macromolecule from these simpler molecules. Now, to do that, what we're going to do is a process called anabolism. So we are going to bring them together, and what we're going to do is we're going to use the first and the fourth carbon hydroxyl group, and we're going to remove water. So what we've done is we've actually had a reaction occur, and this reaction is called a condensation reaction because water is formed as a byproduct. So the water has been removed, and what we are left with is a covalent bond holding the two monomers now together in a disaccharide form. Now the disaccharide, therefore, in this case is called maltose. We've taken the two glucose monomers and we've now joined them together at carbon 1 and carbon 4 with a covalent bond. Now that being held together is called a glycosidic bond. It's called a 1-to-4 glycosidic bond. Now any disaccharide that is formed, i.e. fructose and glucose, will give us the disaccharide sucrose, and galactose and glucose will give us the disaccharide lactose. Any disaccharide that is being held together by a glycosidic bond. So it's important that we note that disaccharides are formed by condensation reactions, and we note that the monomers that are bringing them together is important. So glucose and glucose is maltose, glucose and fructose is sucrose, and glucose and galactose is lactose. So we've taken our monomers, our simple molecules, and we've built them into a larger macromolecule. Now at this point we only call it a macromolecule because we've only joined together two monomers. If we want to look at these monomers and disaccharides and we want to identify if they're present in a solution, we would do what's known as a reducing sugar test. So if we want to do what's known as a reducing sugar test, what we would do is we would be looking at the presence of our monomers, glucose, galactose, fructose, or our disaccharides, maltose and lactose. Now, these are called reducing sugars because they are able to donate electrons. So their ability to donate electrons means that they can, in the presence of a Benedict's reagent, actually change the Benedict's reagent from its negative result, which is blue, into a positive result. So what we would do is we would take a sample of whatever our solution is. We would put in the same volume of Benedict's. We would heat them in a water bath, and if we get a positive result, as you can see here, my blue color has been changed into a yellow color. Well, the positive result is a colored precipitate. Now that colored precipitate can go from anything from green through to yellow, through to orange, through to brick red. Now brick red is the one that indicates a high concentration of reducing sugar. However, the other colors, we are identifying a color, so we are saying that there is a colored precipitate, there is a reducing sugar present, but we cannot guarantee that concentration because we're using a qualitative result. Now, if we're looking at a non-reducing sugar, i.e. sucrose, and we want to identify if that is present, what we would do is take a sample and complete the Benedict's test. Now that should give us a negative result because it's a non-reducing sugar. The sucrose cannot donate its electrons. What we need to do is we need to break this disaccharide down into its two monosaccharides. And we do that by heating it with hydrochloric acid. We need to break that glycosidic bond. When we break that glycosidic bond, we now have our monomers, our glucose and fructose, are reducing sugars, which will then donate their electrons. But we've heated it with hydrochloric acid, so we need to neutralize the sample with sodium hydrogen carbonate before we conduct the Benedict's test again. So if we're using a non-reducing sugar, i.e. sucrose, that's the one you need to know, then we have to do the Benedict's test first, get a negative result, and then we would do the heat with the hydrochloric acid and the neutralization with the sodium hydrogen carbonate before doing Benedict's test again. So in an exam question, it's important you note whether they've told you if it's sucrose, they're expecting you to know that that's a non-reducing sugar, or if they've named a different non-reducing sugar, they will tell you that it's a non-reducing sugar and then expect you to know to use the non-reducing sugar test. So if we then take our disaccharides and we build them up into polysaccharides, the first polymer I'm going to talk about is the one that's found in plants. So starch is a polymer of glucose which is found in plants. It is insoluble, and it is the way in which the plant will store the energy that it will require at a later date. So it's actually made up of two molecules, one which is called amylose. Now amylose is a polysaccharide which is held together with 1 to 4 glycosidic bonds. So therefore it is a unbranched molecule which is formed, but what happens is because of those 1 to 4 glycosidic bonds and their positions, this unbranched molecule will actually form a helix shape. Now that helix shape is stabilized by hydrogen bonds within the molecule. That means that it can be compacted down and is less soluble. It also means that it's very resistant to digestion. So starch is a form where you have your amylose, which is about 10 to 30% of the starch that's present is made up of this amylose, 1 to 4 glycosidic bonds, unbranched molecule which is helix shape and compacted together. The rest of the starch molecule is actually in the form of amylopectin. So 70 to 90% of starch is found to be amylopectin. Now that not only has a 1 to 4 glycosidic bond, but it also has a 1 to 6 glycosidic bond. That therefore means that this is now branched. So for every 20 to 25 units, you will have a 1 to 6 glycosidic bond. So this branching off, this makes it more compact and even less soluble than the amylose. So it's a globular molecule which is held together, more compacted. Now, if we're going to test for the presence of starch, the amylose or the amylopectin, we would do an iodine test. So the iodine test is where you would dissolve the iodine in potassium iodide, and you would then test your sample with the iodine test. If there is no starch present, you would get what's known as a negative result, so an orange-brown colour. If starch is present, either the amylose or amylopectin, that would give you a positive result, which would give you a blue-black colour, which would be present. Now, in animals and in fungi, we have a storage molecule which is called glycogen. Again, it is 1 to 4 and 1 to 6 glycosidic bonds, but this time it's branched every 10 units. So instead of it being every 20 to 25 units in plants, it's every 10 units. That is very important because that makes it a really good compact energy storage because animals are much more active than plants. Now, it is also an insoluble molecule. So because it's an insoluble storage molecule, it means that it does not affect the water potential. It is important that we are not affecting our water potential in animals and fungi because animals do not have a cell wall. Fungi do have a cell wall, however, animals do not. And therefore, we do not want to affect the water potential inside our cells. If we're storing glycogen as this storage molecule, and we want to maintain our cells exactly as they are, we need to ensure that we do not affect that water potential. So by storing our energy in the form of glycogen, it means that we are not going to affect the water potential, and therefore our cells will not take in water by osmosis or be ruptured. So it's a really important way in which we can have this compact, large amount of energy stored in a very small volume. Now, the next thing we need to understand is that it's metabolically inactive. So glucose is our respiratory substrate that we would use, and glycogen is our storage of glucose. So what we need to do is we need to be able to break it down into our glucose molecules so that it can then be utilized through aerobic respiration. So glycogen is a metabolically inactive molecule, so we are storing it for a later date, but it can be broken down quite easily. It can also be added to quite easily, and that therefore means that it is a very good storage molecule. Now, cellulose is another polysaccharide, and it's a polysaccharide that is found in plant cell walls. Now, this one is a polymer of beta glucose. So let's just take a step back and remind ourselves about the beta glucose. Now, beta glucose has the hydrogens below, but what we're interested in right now is that hydroxyl group. Because the carbon 1 and carbon 4 of two beta glucose molecules side by side would be too far apart, what happens is when cellulose is bonded together in a condensation reaction, it needs to be inverted. What we need is for those hydroxyl groups to be on the same level. So what needs to happen is that with every unit of cellulose, that beta glucose needs to be inverted 180 degrees. And that therefore leads to an unbranched 1-to-4 glycosidic bond, but it's a beta 1-to-4 glycosidic bond because of this inversion. So if we looked at our cellulose, which is part of our cell wall in plants, we know that it's inverted 180 degrees every unit. We know that it's unbranched and that it has a 1-to-4 glycosidic bond. We know that it is insoluble. So it's insoluble, and we know that it forms what's called cellulose microfibrils. So our long polysaccharides, the cellulose, small fibrils, will form hydrogen bonds between each other. Now what that does is that makes very long molecules. Then what happens is those microfibrils will then form even more cross bridges, which form macrofibrils. And those macrofibrils will then come together to form cellulose fibers. Now those cellulose fibers have enormous tensile strength. They can withstand the turgor pressure of the water moving into the cell, and that therefore gives the rigidity of plant cells maintaining their shape. And that allows the plants to grow upwards against the force of gravity. So it's important you know about those three polysaccharides: starch, glycogen, and cellulose. Now that is through building those monomers together and those macromolecules being created, but we can also break them down. So if we are going to break down these complex molecules into simpler molecules, what we therefore need to do is we need to do what's called a hydrolysis reaction. Lysis means to split, hydro with the addition of water. So by adding water in, we can release energy by breaking the macromolecule, that complex molecule, down into the simpler molecules, the monomers. So it's important that you know how we can build up anabolism and how we can break down catabolism, and that we understand with saccharides we are either forming the glycosidic bonds or we are breaking the glycosidic bonds. Now, all of these reactions require enzymes to help the process happen at a faster rate. Because we are needing to either form the bonds or break the bonds, enzymes are required. So cellulose, that microfibrils, those macrofibrils, and then the fibers, if we don't have the right enzymes, we actually find it really difficult to break apart the glycosidic bonds, and therefore that forms our fiber, our undigestible molecules that helps with the movement of substances through our digestive tract. So I hope you've liked this video, and if you did, then please click the like button below and subscribe to my channel. Also, do please check out my revision platform, www.aiqchat.com, which will help you with revising for OCR A.